welcome to a new episode of IBD Essentials. I'm very happy and very proud that my long-term friend and colleague Matthieu Allez is today with us. And Matthieu Allez will for sure introduce himself a little bit, but those of you who know him, he is an immunologist, he is a clinician, he was long-term based in Paris, at the unit there, and now recently just moved to Canada. So I'm also interested how this change is doing with him. And again, for the audience who are following us for a long time, you are very appreciated. If you like us, if you have any new topics, new wishes that you would like to discuss, just inform us. You can write your notes, you can like us just on this page, and we will really address this. Matthieu, it's a great pleasure to meet you. And as I already mentioned, we are long-term colleagues and friends, had very nice evenings together, had a lot of activities with ECHO, so we really know each other very well. But I was really astonished because you, as a real Parisian living for all the life I know with you, just being in Paris, moving to Canada, to the new world, and now in Trump's time, maybe even a country which is almost taken over from the US. So how is it going? Perhaps you introduce yourself first.
Yeah.
And then...
Thank you, Axel, for all these words, and it's really a pleasure for me here to come to this interview and to share with you our ideas, our experiences. So, as you know, I was trained in Paris, in Hospital Saint-Louis, and I became head of the department 16 years ago for 15 years. I had also a lab, and as you know, we have been working on IBD for so many years now that we have been following the patients who suffered from the disease, different diseases. And for me, I had the chance very early in my career to get to meet people who were doing research, and I became curious and I wanted to better understand the disease. So I came to New York, I did a postdoc at Mount Sinai, and I came back to Paris and all these stories. And I had the chance to do clinical and research together, and that really fed me. That was my chance, I think, that to try to understand the mechanism of the disease, but also to maybe how to communicate that to the patients, how to improve the management of our patients. That has been really driving me. And about Canada, it's a great experience, actually. When I arrived there 1 year ago, I had the feeling that I was entering in a monastery. I was a little bit alone, but I met the team there, and there is big opportunities because I am at the University of McGill, and I integrated the research institute, and there are researchers in other fields, and this interdisciplinary aspect is for me fascinating because we can really make a group. And we can make a group within McGill, but in fact, what is interesting is to improve the networking with other collaborators in Canada, but also in Europe, all over the world, because I think that's where the research is right now moving, is that we now realize that we can all work together in a way that we can take advantage of the different expertise. So already I spoke too long.
No. It was perfect, and we have a lot of similarities. We both had basic science background and then a clinical background. I didn't move, but ultimately...
But you were trained with by Claudio Fiocchi.
And you moved to Montreal, and Montreal is probably, for a French-speaking person, it's so you're back in France almost.
Oh, it's North America, but it's interesting. There is a French touch, and it's interesting, but it's not exactly France.
Yeah.
It's not Europe.
What I really would like to discuss with you today is the whole area of cell-based therapies, because you, as an immunologist, you know the cells. I've been working on the cells for quite a while, but you have also been instrumental for clinical trials in the field. Most of us will probably remember the ASTIC trial, the hematopoetic stem cell trials, and there is a lot of background before then the trial. and then probably some aspects that are still not perfect and still not the solution. We have CAR T cells in other areas, and we have first results in IBD. So I think this is a very exciting topic, and I really would like to discuss with you. So how did, probably as an immunologist, you moved into the field, but how did you get to the clinical trials, to the aspects, and what do you believe is important?
So it's a long story, and there are many stories, but I think stories are interesting. It's, the first attempt was in France. Marc Lehmann was contacted by Pierre Desromeaux from Lille, and we did a clinical trial on regulatory T cells. And I was very attracted by this idea because I did my postdoc on regulatory T cells. So globally, regulatory T cells are like peacekeepers in the gut. And we know that in IBD, they are not sufficiently activated. So we don't know if the disease is caused by a deficiency in regulatory T cells, but we know that we can improve the course of the disease. We can try to restore the homeostasis in the gut, controlling inflammation. So we did a study on expanding regulatory T cells in the blood and re-injecting the T cells to the patient. It was a phase 1 study, which was quite complicated because T cells were activated by ovalbumin, and we needed to give back to the patient some ovalbumin orally by meringue. That was a funny aspect. We were not sure that was necessary, but we did that. There was interesting signals, but there are still people working on regulatory T cells, even with CAR T cells, actually. But we moved to other ideas, and the story about autologous hematopoetic stem cell transplantation, HSCT, was quite interesting. You remember we had patients who had IBD and who unfortunately experienced another disease like malignant disease like lymphoma, leukemia, or even breast cancer. And those patients got hematopoietic with stem cell transplantation, and autologous. And what is interesting is that many of them had a benefit effect and with long-term remission. So that's why we started to think about doing this procedure to patients who had refractory Crohn's disease. And the difficulty was to select those patients who could be relevant for this treatment because it was not very safe. There were some risk that we have been controlling over time, but some risk. And in fact, the principle is to take, again, blood from patients to expand stem cells. And then when we have these cells, we go back with a very intensive chemotherapy plus depletion of T cells, and we give back the stem cells. And the experience was quite interesting because for many patients, we changed the course of the disease. But not all patients, actually. But the majority of patients had a good response with the treatment. Unfortunately, it is not a cure. And that's what we discovered with that. In fact, the disease come back. But what was fascinating is that when the disease come back, if you take the treatment which were failing before, they work again. Like we had patients who had refractory to multiple biologics. They had autologous stem cell transplantation. They were doing well, but on endoscopy, we saw recurrence. So we started biologics. At the beginning, we used anti-TNF after VEDO. And then the patients responded very well to this drug, which were failing before, like the concept of resetting of the disease.
This was what I was thinking, the reset of the immune system to start new. And initially, I think some of us were believing that this would be sort of a cure because the immune system would be really restarted and resetted, and then it would be much, much better. But as you already mentioned, this is not really true. But I think most of us were really impressed by the initial results from the groups in Chicago, where they treated like 20 patients and 19 were healthy. Looking to the real clinical situation now, we see it's far away from this very optimistic data, or I'm incorrect?
Well, in ASTIC, actually the trial was negative because we had a very stringent, very ambitious endpoint, which was remission without treatment, without any signs of inflammation. And very few patients reached this endpoint. But as I said, what we found is that many patients benefited, and it was kind of resetting. And you say a resetting, what we have seen, in fact, that you reset the adaptive immune response, like memory, But in fact, that does not cure the disease. So there are other mechanisms driving the recurrence of the disease that we need to identify. And it's a beautiful model to better understand the pathophysiology of the disease. And unfortunately, in ASTIC, there was one death, sorry. And we also had other experiences. So it's not completely safe. And the problem by doing that is that we had always new biologics, new therapies coming, and then we were talking about what should we do for the patients, and we always try the last biologic. So that's what makes this story a little bit difficult, and it is reserved for very refractory patients. But still, I think it's interesting.
Yeah, but this brings me to one important aspect, selection of the patients.
Yeah.
You said already highly selected patients.
Yeah.
Which patients are appropriate, for instance? This needs to be discussed. And you also mentioned the endpoints. So if you have patients where no therapy works and you are desperate, so even if you don't get a cure, but a therapy would work again. So if you have not the ultimate endpoint of cure and healing, this would be also appropriate. So I think this is important for clinicians at least.
Yeah. So selection, at the beginning, we had a committee and we discussed all cases in ASTIC actually. And after that, we did that in each country. So we established that you need a multidisciplinary meeting to validate or not the indication of autologous stem cell transplantation. Actually, in Paris, we set up that. And the patients who are good candidates are probably those who have refractory disease, which cannot be improved by surgery. And it's important to have surgeon within the room, especially when you are on small bowel disease. And those patients who are at risk of a poor outcome, when there is a risk of short bowel syndrome, there are patients who are very high risk, in fact. On the other side, we saw no effect on perianal disease. So patients who have refractory perianal disease are probably not good candidates for autologous stem cell transplantation. And there are still people doing it in the world right now. There are some centers still analyzing that in multiple cities, like in New York, like in multiple centers in Europe. And now we have new cell therapy, which are coming, and as you mentioned before, CAR T cells. And I think that's quite interesting with the New England paper published by the German team on the UC patients who had ulcerative colitis patients, refractory, young female. Actually, she could have had surgery, but they decided to do it, and that was a fantastic experience. And interestingly, when we talk about selection, what is interesting is that the CAR T cells which were used were targeting CD19, which is targeting the B cell pathway, but not CD20. So there are small differences, but that's quite important. And it was in a patient who had autoantibodies. And that's something that we discover now in the recent years. We start to see that, in fact, there is some autoimmunity in inflammatory bowel disease, something that we were not, we were excluding actually 10 years ago. And now we have data showing that some patients have an autoimmune component and they may be good candidates for this kind of therapies.
But you bring it back again to the question, which is the right cell. And you mentioned CD19, CD20. For most of us, I think IBD are still T-cell driven diseases and not B-cell driven diseases. And now there is a shift, and you already explained a little bit that this may be the case, and make it even more complicated because you mentioned regulatory T cells where you started, and modification also to get regulatory properties back could be also an approach. So how can those who are not really immunologists deal with these different aspects? How
well, the reality is complex. That's what we could summarize. It's difficult to know, but we have learned a lot about T cells. And in fact, there is a big link between CD4 T cells and B cells. They collaborate, they exchange information. So we can find signals of autoimmunity on T cell actually. And we have done that. We have shown that some patients share the same T cell repertoire, like public repertoire. We don't know what are the antigens recognized by these T cells, but they are there. And we need to work on it. And on the other side, there are some clonal T cell expansions, which are private, which play a role, I think, in refractory disease. So it's another aspect. And with targeting CD19 B cells, we are targeting adaptive immune memory, and we are targeting some mechanism which may be very early. And if we identify those patients who have an autoimmune component, because one aspect that we should say, I think we all have these ideas together, is that there is heterogeneity in IBD, and we have probably multiple subtypes. So the idea is that maybe we should define what is the best strategy for the subtypes that we would be able to identify. And I think that's coming with all the research on autoantibodies and T cells, adaptive immunity. And besides, there is innate immunity and pollutants and microbiome, and so that's quite complex. How do you see that? This aspect, Axel? For me, we will make progress if we are able to identify subtypes of disease.
No, I think this is a very important aspect, and I'm completely in line with you. I believe we do not have one IBD, we do not have one Crohn's disease or one ulcerative colitis. For me, it was never understandable why some patients will develop fistulizing disease, or some with Crohn's will have upper GI involvement, some will have strictures and no fistula. These must be different diseases just due to the similarity in the histopathology initially and some inflammation in the mucosa. It was lumped together, but personally, I do not believe that this is one disease. But this brings me and makes it even more complicated. We are discussing now all these techniques. Which are the patients that should undergo this? Is it only for Crohn's disease? Is it for colitis undefined? Or is it also for ulcerative colitis, where a lot of people could say, we have surgery as cure, and this will bring us to the next problem then, the cost of all these treatments.
Yeah.
So could you first, for our listeners, define who would be an appropriate patient, and would it be the Crohn's patient or also the ulcerative colitis patient, or those with extraintestinal manifestations, even if you take out the gut and they still have pyoderma or something?
Well, I think, difficult. But that's funny because the German team in Erlangen, they published they choose to treat a UC patient who had refractory. She refused surgery. She had autoantibodies again. So they selected. So I think for me, an expensive treatment should be applied like a little bit like the story of autologous stem cell transplantation in refractory disease. And then we need to see to evaluate the efficacy. And I guess that in the future, if we have a game changer, a treatment which completely changes the history of the disease, maybe in terms of cost benefit, economically, that should show that there is a benefit to do it. And then we will have to define, I think, over time, how to select the patient. And that's quite interesting because the selection will be based on multiple aspects. Us, as clinicians, we are concerned about those patients who have a poor prognosis, exposed to surgeries and so on. There is quality of life, all these aspects of, we have treatment, but if we can improve, today we have many treatments which are efficient, right? And we are making progress on combining multiple biologics. So that's a big problem in the field, is that we have more and more strategies which are available. And how do we choose?
Yeah.
And even for research, where is the way? So I think that's a good idea with cell therapy probably is to start with refractory disease where other options fail because of the cost, because of this. That would be my...
And then I push you hard again. So you mentioned they did ulcerative colitis. You were just moving a little bit around. You were about the refractory, and I would say ulcerative colitis. Our surgical colleagues tell us there is a cure, and we know that it's not a cure. But so would you also consider this for UC patients, or is it more for the undetermined colitis and for the Crohn's disease patient?
Well, the truth is that sometimes, you know that, patch surgery can be fantastic, but sometimes it's not. We have patients who have, in fact, a disease which looks like Crohn's developing on the pouch and so on. So we have not completely solved the problem. And of course, we have patients with a pouch with a quality of life, like 3 stools per day, but we have other patients who are more difficult. And then it's difficult to, I mean, it's a difficult decision, surgery in ulcerative colitis. And now we have more and more treatments, and then that may increase the risk of malignancies, actually, because before we were doing surgery and so on. So we have to balance the different options. I still think when you see, for example, in autoimmune disease, the results on lupus or multiple sclerosis, on myasthenia, myositis, that's quite impressive. They almost cure some patients. And let's see there is one case of UC, we will see how Crohn's work, and then it will depend on the efficacy, cost.
I think one important issue we should discuss in the end is safety. So you mentioned safety, the death in the ASTIC, so hematopoetic stem cell therapy. How is the safety issue in CAR T cell therapy? And final question also in this respect, the resetting of the immune system, somehow that treatments will work, because I'm still not optimistic that it will be a lifelong cure. It will be similar in a way.
Yeah, well, probably. And I think that even if we do CAR T cells, maybe there would be strategies after to look at recurrence and how to manage if there is recurrence and so on. The safety of CAR T cells targeting CD19 seems to be good. However, when you look at the patient, she has hypoglobulinemia. And there is no consequences, but we know that hypoglobulinemia can expose to some infections. So that's one aspect. So we need more data, we need more experience, long-term data. When we assess safety, short-term is not best. We know very well that. I think so far, and I'm not an expert in that, the experience is quite acceptable. It's better tolerated than autologous stem cell transplantation, I'm quite sure. But we need to be careful on the long term and so on. So that's one aspect why probably we need also to move slowly. We should not. It may be a revolution, but we don't know. Also, we don't know from other fields of research. We can also have other revolutions, right? If you find some role of specific microbes, and we are still working on it, for specific subtypes of disease, that could be the solution for some patients.
No, I think this is really perfect, and you opened several new roads which need to be followed, and I think we should really have a follow-up podcast because I could also have challenged you that all the immunologists are not the correct people. It's a barrier problem, and we could talk about the epithelial cell. But this would lead far too far.
No, but it's very linked, and probably if you analyze subtypes, we may find different types of disease where specifically epithelial cells or some subset may be key players. But who knows? That's difficult to establish.
So Matthieu, thank you so much.
Thanks,
it was a real pleasure to discuss with you. And to our audience, I hope you liked this. You may like us. You may ask for new episodes, new aspects. We were happy to entertain these. Thank you so much for visiting us.